Bismillahirrahmanirrahim. Assalamu alaikum students. Today I will be discussing with you people an important topic of microbiology, that is complement system. Students, by definition, complement system is a group of 20 different types of proteins which are present in our serum. Okay, 20 different types of proteins present in our serum. Okay students, why these proteins are called complement? This is because these proteins, they augment, they increase, or we can say that they help in the immune system of our body. Okay students, second point, complement system is a part of innate immunity. Complement system is a part of our immune or innate immunity. These are two characteristic points regarding complement system. Okay, now students, how this complement proteins they are activated? Students, these complement proteins, they are present in our serum, and how they are activated? They are activated by three pathways. Number one, students, first pathway is classical pathway. Classical pathway for activation of complement proteins. Number two, alternate pathway. Alternate pathway. And number third, lectin pathway. Okay students, these three pathways, they are responsible for activation of complement system proteins. Students, classical pathway, it is antibody dependent. That is, it always requires the presence of antibodies for its activation. Classical pathway, it is activated by presence of antibodies. These antibodies, they are either in the form of immune complexes or independent or isolated antibodies. Number second pathway is alternate pathway. Students, alternate pathway and lectin pathway, both of these pathways are independent of antibodies. That is, they do not require the presence of antibodies for their activation. They need only offending antigen for their activation, and this offending antigen, it may be simple bacteria, viruses, or even the endotoxins produced by different strains of bacteria. Okay, these are the three pathways which activate the complement proteins. Now students, whatever the pathway is, there are three main points in all these three pathways. What are those 3 points? Number 1, formation of C3 convertase. Formation of C3 convertase enzyme. Number 2, formation of C5 convertase. And number 3, formation of membrane attack complex, MAC. MAC stands for membrane attack and complex. Okay students, these 3 points, they are common in all the 3 pathways of complement activation system. That is, number 1, formation of C3 convertase enzyme, formation of C5 convertase, and number 3, formation of membrane attack complex. Okay students, these 3 components, they were common in all the 3 pathways. Now we move to C3b molecule. Students, C3b molecule is the main product of all the 3 pathways. That is, C3b complement molecule protein. It is formed as a result of activation of either the complement system, classical pathway, either the alternative pathway, or either the lectin pathway. It is the main point, main molecule formed as a result of these 3 pathways. C3b molecule. Students, what is C3b molecule? C3b molecule, it is also a complement protein. Okay students, and it is the main protein which becomes attached on the surface of offending antigens and makes it easier for the phagocytic cells to destroy them. Okay, C3b molecule, it is a complement protein. It becomes attached on the offending antigen, and as a result, this offending antigen becomes easier for phagocytic molecules to destroy them. What are phagocytic molecules or cells in our body? Macrophages, neutrophils. These are examples of phagocytic cells in our body. So C3b molecule. Now students, there is another structure which is called membrane attack complex. Membrane attack complex. Students, what is membrane attack complex? In simple words, membrane attack complex is a combination of different types of complement proteins. What are those complement proteins? C5b, C6, C7, C8 and C9. Students, these 5 complement proteins, they join together to form an effector protein which is called membrane attack complex. I repeat, what is membrane attack complex? Membrane attack complex is effector protein of immune system formed by a combination of C5b, C6, C7, C8 and C9 complement proteins. These 5 complement proteins, they together make up to form effector protein called membrane attack complex. Okay students, now what is the function of membrane attack complex? Students, membrane attack complex is formed as combined activity of classical pathway, alternate pathway plus lectin pathway. These 3 pathways, they result in the formation of membrane attack complex. Okay, now students, the main function of membrane attack complex is cell lysis, that is breakdown of cells. How breakdown of cells is caused? Membrane attack complex creates very fine pores in the outermost wall of the cells, and as a result of these pores, the cell wall is destroyed and cell constituents are leaked out. Okay students, the membrane attack complex, it causes cell lysis by creating small pores in the cell wall. Students, membrane attack complex causing cell lysis, it is a typical example in mismatch blood reactions. When one when donor's blood is transfused to the recipient without proper cross matching, membrane attack complex is formed as a result of complement activation, and it causes breakdown of RBCs. As a result, there is mismatch, cross-match reaction in the human body. Okay? Now students, we move towards biological role of complement system. Biological role. Biological role of complement system. What complement system performs in our body? You can say functions of the complement proteins in our body. Number one, students, opsonization. Opsonization. Students, opsonization means to help in the killing of offending antigens. How help? To make offending antigens more easier for the phagocytic cells to destroy them. What are those complement proteins? C3b. Okay students, C3b becomes attached on the surface of offending proteins like bacteria, viruses, and as a result, the phagocytic cells, they can destroy the offending antigens more easily. Okay. Number second function of complement system is chemotaxis. Chemotaxis. Students, in very simple words, chemotaxis means to attract, to accumulate. These complement proteins, especially C5a, okay, and combination of C5, 6 and 7 complement proteins, they cause chemotaxis of neutrophils at the site of injury. I repeat in other words that these C5a complement proteins and combination of C5, 6, 7 proteins, they cause recruitment, or they attract neutrophils at the site of injury. Students, neutrophils. Neutrophils are main cells which participate in the process of acute inflammation. Okay? Opsonization, chemotaxis. Number third function of complement system is Anaphylotoxins. Students, complement proteins like C3A, C4A and C5A. These three complement proteins, C3A, C4A and C5A, these three complement proteins, they destabilize the mast cells. As a result of this destabilization, the mast cells, they release certain types of chemical substances into the blood. These chemical substances released by mast cells are called anaphylotoxins. Students, one of the example of anaphylotoxins is histamine, serotonin, there are 10 to 12 different types of chemical substances which are called as anaphylotoxins, and these anaphylotoxins, they are released in the blood when mast cells are destabilized. How mast cells are destabilized? These mast cells are destabilized by complement proteins C3A, C4A and C5A. Now students, what is the effect of release of anaphylotoxins in our blood? Students, their main function is smooth muscle contraction. Smooth muscle contraction. And number 2, increased vascular permeability. Students, these are the two main effects of anaphylotoxins. Smooth muscle contraction. Mainly, smooth muscles are located in our respiratory tract. And when these histamine, serotonin are released in the blood by the mast cells, there is contraction of smooth muscles of our respiratory tract. As a result, the patient feels difficulty in breathing. Second is increased vascular permeability. Students, it means that blood present in our vascular system, it leaks out of the blood vessels, and as a result the patient feels edema or swelling at the site of injury. Okay. Students, we will study anaphylotoxins and their role in subsequent topic of hypersensitivity type 1. We will discuss it later in detail. Now, the fourth biological role or function of complement system is cytolysis. Cytolysis means breakdown of cells. As I have already told you that membrane attack complex C5b 6 7 8 and 9, membrane attack complex causes lysis or breakdown of cell wall of different cells. Okay, the last function of complement system, complement proteins, is antibodies production. Antibody production. Students, complement protein C3b, it becomes attached on the offending antigen, and as a result B cell, B lymphocytes, they recognize C3b through its receptors, and as a result there is production of antibodies from B cells. Okay, so we discussed 5 different biological roles of complement system. I repeat: number 1, opsonization; number 2, chemotaxis; number 3, anaphylotoxins; number 4, cytolysis; and number 5, antibody production. Okay, students? Now, students, we discuss clinical aspects. Clinical aspects of complement system. That means, what is the importance of complement proteins in our body? Number 1, students, deficiency of C5, 6, 7 and 8 proteins in our body, it leads to more risks of Neisseria bacteria infection. Okay, it means that if these complement proteins 5, 6, 7 and 8, if they are deficient in our serum, then patient is at more risk of developing Neisseria bacterial infections. Okay, number 2, deficiency of C3 complement proteins. Students, if C3 complement proteins are deficient in our body, then we are at increased risk of developing respiratory infections. Okay? Pyogenic sinuses infection. Okay? Deficiency of C3 protein, it leads to respiratory infections or pyogenic infections of our sinuses. The air pockets. What are sinuses? Sinuses are air pockets which are present in our bony skull, in the